In this part, now Mr. Hugo Pereira will give us value information about industrial scale production of microalgae. But before his speech, I would like to give some information about him. Mr. Hugo is the general coordinator of the GreenCoLab, a novel collaborator platform between research and industry, whose research and innovation agenda is based on exploring macro and microalgae as essential components for different industries. Previously, he was a collaborator in the March Biotech group and in the RD departments of Necton and all microalgae. He completed his PhD at the University of Algae on the production of biomass at industrial scale, as well as on the establishment of the microalgae-based biorefinery for Tetraselmis for different biotechnological applications. Hugo has 60 publications in peer-reviewed journals, 4 book chapters, participated in various national and international projects related to algae biotechnology, and he is coming to the stage with the speech title, Production of Tetraselmis at Industrial Scale and Development of a Novel Biorefinery Approach in 4 different biotechnological applications. The floor is yours.
Okay, good afternoon, everyone. I'm Hugo from GreenCoLab, and as our colleagues said, it's a collaborative laboratory that aggregates different companies and the research institutes working in algae biotechnology in Portugal. And today I will try to give you an example of a work conducted with different associates, namely the production of Tetraselmis at industrial scale and the establishment of a novel biorefinery for different biotechnological applications. The presentation is divided in 4 main sections. First, I will give you a brief introduction about Tetraselmis UTP4. Afterwards, the work conducted at all microalgae in the industrial production of these species. Then a biorefinery concept, a novel concept that was performed at the Center of Marine Sciences in the south of Portugal. And finally, a feed trial that was conducted with the company Sparus. So, as everyone knows, bioprospection is a very important process of algae biotechnology. In this context, in GreenCoLab, we sample different water courses and try to promote the isolation of novel strains in solid or liquid media. And in this way, we can have newly isolated autochthonous strains that are already adapted to our growing conditions. We do this using flow cytometry. I will not go in detail how the flow cytometry operates, but we use a combination of different features that the cytometry can read, such as the relative cell size, the inner cell complexity using the side scatter detector. We also use the autofluorescence of the pigments, such as chlorophyll and phycoerythrin, and in some cases, we stain using exogenous dyes, as for example, body P, that can stain for the lipid bodies of the cells. And using a combination of different parameters, we use the fluorescence-activated cell sorting to separate new populations. In our case, the flow cytometry that we use can separate 50,000 cells per second with a purity and recovery higher than 99%. So this is a way to expedite the traditional streaking technique. Here is an example of the flow cytometry approach that led to the isolation of this new strain, and you can see here with the letter C, Tetraselmis UTP4, and the gating procedure that was conducted. The strain was identified by means of 18S rDNA sequencing. We knew that it was a Tetraselmis strain, but this analysis confirmed that it did not belong to the traditional Tetraselmis shui and Tetraselmis shuesica that are widely studied. It clearly belonged to Tetraselmis striata convolute clade. So in summary, strain CTP4 is a marine chlorophyte. It was isolated in the lagoon of Ria Formosa in the south of Portugal. It's a fast-growing and robust strain that can tolerate wide environmental conditions. It's an autochthonous strain already adapted to the surrounding environment, but for us and for a biorefinery, the most important feature is that it can accumulate high lipid amounts under specific culture conditions. And here, just as an example, under nutrient repletion, you can see a stain with body P. We have about 10% lipids, but manipulating the culture conditions using nutrient depletion and the combination of abiotic factors, we can increase significantly the lipid content. We can reach now about 50% of the total dry weight in lipids. So because of the potential of this strain, we were invited by all microalgae to test the production of the biomass in their production facility. All microalgae is an industrial facility that produces autotrophic and heterotrophic biomass and has a production capacity higher than 1,300 cubic meters. So when I arrived to all microalgae, I only had one agar plate with the strain. So the first thing I did was a scale-up. So I passed from the agar plate to an Erlenmeyer, to vertical columnar lids, green wall panels, tubular photobioreactors. And all this process took me about 8 weeks to reach the industrial scale systems that you see here in this picture. We conducted 2 trials at all microalgae. The first one was conducted in the autumn-winter season. We used 2 photobioreactors, one with 35 and another with 100 cubic meters. The systems were operated semi-continuously for 60 days, and the cultures were harvested 4 times. We had a standard production pipeline, so we produced the biomass in the photobioreactors. We harvested by ultrafiltration, and we processed using a spray dryer to obtain fine powder. Here you can see the results of the first trial. The trial was conducted between the 17th of October and the 14th of December. Here the temperature on site, also the radiation. And here you can see the growth curves of both photobioreactors. And it was very interesting to see that the 100 cubic meter, we always had higher growth of the culture in this system, and this is super important from an industrial perspective. Our volumetric productivity in the winter was up to 0.09 grams per liter per day, while the aerial was between 10 to 20 grams per meter square per day. It's also important to highlight that during this trial, we were able to keep monoalgal cultures during the 60 days, although we had some contaminants such as amoebas, ciliates, verticillas, and so on. Then in the summer, we came back to do the second trial. In this one, we used the biggest photobioreactor present at the company with 150 cubic meters. Again, we operated semi-continuously for 60 days, and the cultures were harvested 5 times. We used the same production pipeline, but in this trial, we tried to address the sustainability of our water source. So we decided to continuously recirculate the culture media after the membrane filtration. So we never used fresh water. We were completely recirculating the permeate back into the photobioreactor. Here you can see the growth curve of the trials. We reached much better results during this season, reaching a biomass productivity of 0.15 grams per liter per day, and the aerial productivity higher than 32 grams per meter square per day. It's also important to say that in these 60 days, we produced more than 1 ton of biomass of Tetraselmis UTP4. After the biomass production, we tried to take advantage of a unique feature that Tetraselmis has. The cells are very big and they can settle. So we developed a pile scale system to harvest the biomass. We do this by a 24-hour sedimentation step using gravity drainage only. In this way, we can remove 95% of the culture media, while 5% of the culture is retained in the tank. So afterwards, we are able to remove this concentrated culture, and this represents a biomass efficiency of 96%. However, some of the biomass, I'm sorry, I need water. However, some of the biomass still stays in the bottom of the tank, and we are able to recover this biomass using a scraping system, which has a dry weight of about 273 grams per liter. So after the biomass production, we went for the biorefinery of the biomass. So we did this using an ethanolic extraction procedure. Here is the characterization of our ethanolic extract. You can see on the right and the chromatogram for the oil. We have a high amount of triacylglycerol, monoacylglycerol, and phospholipids. And afterwards, we developed a liquid three-phase system to partition this extract into different fractions. And we were able to divide this extract into a non-polar fraction, a colloidal fraction, and a polar fraction. Still, we have our spent biomass. So from one biomass, we were able to generate 4 different bioproducts. Then we decided to try to valorize these fractions. On the non-polar fraction, we made biodiesel, that you can see in this picture. We also assessed the potential for edible oils. The colloidal fraction is rich in phospholipids and glycolipids, and we studied as a source for human and animal nutrition, whereas the polar fraction, rich in polysaccharides, was upgraded to bioethanol, and we are studying the potential of oligosaccharides as a feed additive. Nevertheless, I will now just focus very quickly on the spent biomass. So to valorize this biomass, we decided to conduct a trial in Sparus aurata. So we decided to replace 10 and 30% in the feeds of Sparus aurata at the expenses of soybean. But why do we want to replace soybean? Soybean is a high productive crop, has a low production cost. It's considered high quality protein with a balanced amino acid profile. Nevertheless, soybean has some drawbacks. It has a high demand for water, has some known growth inhibitors for the fish, such as phytic acid. But nevertheless, the most worrying is that soybean is the main driver of world deforestation in third world countries. And 60% of this deforestation is linked to soybean and derivatives. And the EU is the main consumer of these products. So finding new sustainable alternatives are key. In this context, we decided to test the biomass as a fish ingredient. Here you can see the biochemical composition of our ingredient compared to the soybean meal. You can see that the products have a similar profile. And then we formulated 2 feeds. So a feed containing 10% soybean meal and a feed containing 10% of our microalgae diet. You can see here some adjustments that were made to compensate possible nutritional imbalances. And afterwards, after the formulation, we went to manufacture the feed. So we weighed the ingredients, we mixed and milled them, we added the fish oil, we performed the low shear extrusion, and after drying and sieving, we had our product packed. So afterwards, we went to the experimental room of fish trials. We had 3 tanks with our control soybean diet and 3 tanks with the microalgae-based diet. We used groups of 30 seabream with the initial body weight of 6 grams, and the diets were tested over 60 days. The fish were fed 3 meals a day. They were hand-fed to society, and we performed the analysis of different parameters, such as the fish performance, the weight gain, specific growth rate and food conversion ratio, and the digestibility of protein, fat, phosphorus, and energy. Our results show that the fish had a 3-fold increase in their body weight during the experiment. We did not have any differences in the growth, in the final body weight of the fish. One interesting result was the lower voluntary feed intake of the microalgae diet. This is important, meaning that we gave less fish to obtain the same dry weight. Nevertheless, although the differences are significant, they are not very big. And regarding the specific growth rate and the food conversion ratio, they were similar between both diets. Regarding the digestibility of the diets, here we had very good digestibilities, similar between both diets, more than 90% for the proteins, fat, and energy, and close to 70% for the phosphorus. regarding the digestibility of the ingredient, and here is when we incorporate 30% of microalgae biomass in the diets, we had also very good results. We have very high digestibility for protein, lipids, and energy, but lower for phosphorus, because algae have polyphosphates that are not well digested by the fish. So in conclusion, strain CTP4 was successfully grown in an industrial scale production facility. We were able to keep our cultures monoalgal during the whole cultivation. Different bioproducts were established using this biorefinery approach. The defected biomass can be a sustainable alternative to replace soybean in aquafeeds. Nevertheless, we are still conducting our life cycle analysis, and this is a very complex process because we have many other works that are not here presented. So we are trying to integrate all of this and now trying to make the LCA to really see if the process is sustainable or not. So I would like to thank you all for your attention. If you have any questions, I'll be glad to answer.
Thank you for your contribution. If you have any question, we can continue with the questions. Yeah.
For your nice presentation, I have a question for biorefinery part. After you extract the oil, the lipid part from the microalgae, did you check the phase state of the oil after removing the solvent? Sometimes it is a very viscous oil. Did you have the same case in your?
We have the same issue. And the oils of microalgae, they are not in a liquid form, and they have a lot of compounds that are not very viscous, so it's always attached to the balloons, and this is a very tricky part to work with it. But when we do this liquid partition, then they become much better.
Okay, thank you.
Hugo, thanks for the presentation. Only for curiosity, I would like to ask, you mentioned about the sedimentation in the sedimentation tank for 24 hours.
Yes.
Only thinking about the Portugal and then the weather conditions, have you checked within the 24-hour interval any effect of sedimentation on the biomass content?
Yes. To be honest, we have not checked before and after the sedimentation. Our concept was to try to settle during the night period because the temperature is colder, so we could maintain theoretically the biochemical profile. It took a little bit more, and we repeated, but knowing the biochemical profile of Tetraselmis, and we analyzed it several times, it's quite similar. We don't see significant differences. But this was done not under summer conditions, so there can be much more trickier to have exactly the same profile. And also bacteria growth. We need to further develop on that part.
Thank you very much for a really inspiring talk. So I've got a practical question. So you've got this huge installation, if I remember correctly, 35 times 150 cubic meters.
35,000.
Yeah?
No, sorry, 35 cubic meters.
No,
The reactor or the facility?
The whole facility.
Yes, more than 1,300 cubic meters.
That's wonderful. So how do you keep it? You don't kind of take care of any sterility, right? You kind of work with a microbiome together with the algae, or you mentioned that there were some amoebas, right? Some bacteria, right? Yes. That's not a problem with the growth of the algae there?
Yes, I've been in all microalgae for 1 year. I know well the company. They try to sterilize in the best way they can. So when they remove the culture, they clean the tubes, the piping, they add the bleach, so we try. But nevertheless, the contaminations always arrive. It's a too big system. It's completely impossible to control and have a sterility.
Okay, so that follows the next question. So how long is the whole, can you sustain the one cycle, culturing cycle, to make it you know, sustainable? Of course, that's probably part of your LCA analysis, isn't it?
And
Because you want to
it's-
know how much.
Yes, so for
energy you
Tetraselmis,
put-
they
yeah.
really, we were able to keep for a long time, but it's really dependent on the operation. I can tell you that sometimes it can last for some months, sometimes in 2 weeks we have contaminations. And this has multiple factors.
So that would be an
Can
important
be
thing
a
to
simple
control.
operation that went wrong, can be the water batch that has it was not well sterilized, so it's really a challenge. And all industrial operations at larger scale, contaminations are something that we need to cope with them. We need to control them. They will happen. We just need to know how to manage them and have best practices
Okay,
to
sure.
overcome that issue.
What about the CO2
Yes.
source? Do you add any CO2
Yes, of
into the
course.
whole system? And what is the source of CO2?
We buy it from the national company. That,
Right, so it's like from the
it's not
bottles.
national, sorry, it's international, but the biggest supplier, it's pure CO2, 99.9.
And did you check? Did you check how much you can push into high CO2 concentration? I'm thinking about linking your elegant facility with some, let's say, point sources, for example, CO2 point sources.
But if the contamination arises from the CO2, is that the question?
No. How much CO2 can they be still growing very well in the presence of what concentration of CO2?
That's a tricky question because some algae like pH 7, and this is all about how carbon is mixture in the water. But for instance, Tetraselmis, and we published that, grows much better at pH higher than 8. It's a marine strain. Normally freshwater strains, we grow them at 7, 7.2. Marine strains between 8.5.
Depends on the
The
pH
idea
tolerance.
is really to make a steady system where we define a pH value and then we just control by injection points when the pH rises above that value.
Okay, it's wonderful to see it from this industrial point of view, you know, because
Thank you.
yeah, it's really good stuff.
Thank you again for your important contribution.